Sorry about that break in conversation. So we have a 4-minute video before this one, but I'm going to continue here with the basic configurations of the negative feedback loop. So a negative feedback loop can be configured in two ways. One is a physiological response-driven loop, which is observed in endocrine glands that control blood glucose and calcium and phosphate and blood osmolarity and volume and sodium and potassium and hydrogen levels. And so basically this is the example I just gave to you, where the physiological response is what negates the secretion of the hormone. So after the hormone stimulates a physiological response, a change in physiology will negate the production of the hormone. And you can see that right here. So the endocrine gland produces the hormone targets the organ stimulates a physiological effect such as blood glucose levels, and the blood glucose levels will stimulate or lead to a negative feedback on the endocrine gland. This is a physiological response driven loop. Then we have an endocrine axis driven negative feedback system. We're going to talk about this in the next lecture, but we have what we call the endocrine axis. The endocrine axis usually comes in three levels. We have the hypothalamus, we have the pituitary, and we have what? We have the target organ. These are so the regulation of the hormone that's produced by the target organ is in three levels: hypothalamus, pituitary, and the target organ. And because of this, we know that the hypothalamus produces a releasing hormone, which then stimulates the pituitary gland, which then produces what we call a tropic hormone. We're going to talk about the tropic hormone in the next slide. And the tropic hormones are then going to stimulate the target organs or the peripheral endocrine glands, which then make the hormones, and then the hormones will lead to physiological effects. But in this case, please observe what I'm about to say. In this case, the effects or the negative feedback is stimulated by the hormone produced by the target organ. Okay, the hormone produced by the target by the endocrine gland is what stimulates or inhibits the production of the other levels of the hormone. So this is called the endocrine axis driven loop. All right, the endocrine axis driven loop. All right, let's talk about control of hormone release. So hormones can be controlled. Like I said earlier, hormone is produced and is tightly regulated in the body. Okay, and there are three types of hormone control. Okay, we have the neural, we have the hormonal, and we have the nutrient. Okay, the neural, the hormonal, and the nutrients. Now, there are organs that have all three levels of control. And for instance, the neural. Let's talk about the neural real quick. In the neural, we have the parasympathetic nervous system, sometimes, often controlling the secretion of hormones. We've seen this in organs such as the pancreas, where insulin is made. We also see it in the adrenal glands. Okay, this is the neural control of hormone release. Then we have the hormonal control of hormone release, and that's where one hormone made by one gland stimulates the production of another hormone made by another gland. So hormones stimulate hormones. So we have hormone 1 targets gland 1 produces hormone 1, which then targets gland 2, which produces hormone 2, which then targets gland 1. So this is called hormonal control of hormone release. Then we have nutrient control of hormone release. And this is a physiological response type where a nutrient, like glucose, is able to affect the production of a hormone such as glucagon or a hormone such as insulin. This is the nutrient control of hormone release. So Two words that often confuse me, students, everyone out there, are tropic and trophic. Tropic and trophic. Tropic hormones are hormones that target other endocrine glands. So when you have a hormone that targets another endocrine gland, that's called a tropic hormone. Now you're going to see that the pituitary gland produces a lot of tropic hormones because they target other glands like the adrenal glands. Okay, and then we also have what we call trophic hormone. A trophic hormone is a hormone that has a growth effect, either hyperplasia or hypertrophy, on the tissue that's being stimulated. So remember, we talked about hyperplasia and hypertrophy in the last lecture. So a trophic hormone will increase the size typically of the organ that's being targeted. Okay, so we have tropic and we have trophic. This is a great exam question I will ask you. And a hormone can be tropic and trophic at the same time. Okay. So let's talk about something even more exciting. Let's talk about the classification on based of hormones based on chemical nature. And please spend some time reading this because I'm just going to go through it really quick because I know that you have your textbooks and I know that you are diligent in studying. So hormones can be classified in different ways. Especially we'd like to classify them based on their chemical nature. Okay, we can discuss hormone based on their synthesis, their storage, their release, how they're carried in the blood, what the half-life is, what the mode of clearance from the blood is, and the cellular mechanism of action. So let's look at the different types of hormones based on classification. The first type of hormone based on classification are the proteins and peptides. The proteins and peptides. And these are made primarily of amino acid sequences. Okay, they're made of amino acids, and some of them are modified by glycosylations, and some of them are modified in the Golgi apparatus by what we call post-translational modifications. So it's after they're made, after they're translated in the endoplasmic reticulum, you're passed to the Golgi apparatus where they are changed and modified and added either glucose subunits to them. Okay, and these are protein hormones are made of amino acid sequences. Initially, they are made as what we synthesized as what we call pre-hormones or pre-pro hormones. Oftentimes, they're actually synthesized as a pre-pro hormone, and then that's cleaved into a pre-hormone, and then that's cleaved into a hormone. Other characteristics of proteins and peptides include the fact that they're stored in secretory vesicles. So they're stored in vesicles, and then you have the hormone there. And they're regulated at the level of secretion. Okay, they're regulated at the level of secretion, which means that the production of the hormone is typically regulated at the level of secretion. And that means, for instance, when you need insulin, it's already pre-packaged in a vesicle. That's what it means. Insulin is already pre-packaged in a vesicle. Now you can have more synthesis of new insulin, but by the time the stimulus gets to the target organ, we have insulin already pre-packaged. That's what it means. And they often circulate unbound. Okay, sometimes we have what we call binding proteins. Binding proteins, they function to extend half-life of hormones, and also help in storing hormones for the future. But proteins and peptide hormones do circulate mostly unbound. They're hydrophilic, they like water, and transmembrane, and they use transmembrane receptors. They use transmembrane receptors, which means if you have a cell right here, the receptor for these hormones is going to be on the outside of the cell and not on the inside, as we're going to talk about later. So these are called transmembrane receptors. They have a relatively short half-life of about 4 to 40 minutes. Okay, this picture right here shows the typical synthesis of a peptide or protein hormone. So in an endocrine cell, we have them synthesized as a pre-pro hormone, and then the pre-pro hormone is then repackaged as a pro hormone, cleaved into a pro hormone. Then the pro hormone goes from the endoplasmic reticulum into the Golgi apparatus when it is made into the hormone, and then the hormone is packaged in vesicles and then released when the stimulus come in. So please remember these steps. We have pre-prohormone, prohormone, hormone, and then secretion. And a few examples of hormones that fit into this category are insulin. We have parathyroid hormone. These are peptide hormones. Okay, so I want you to see if you can find other peptide hormones that are available that you can discuss with me. Okay, we have glucagon, by the way, lest I forget, and a few others like that. Okay, other characteristics of protein and peptide hormones is that they're stored in membrane-bound vesicles. I said that already. The examples can be FSH and LH, but these are examples that are post-translationally modified. Okay, their glycoprotein hormones. They're post-translationally modified. We have FSH, we have TSH, and we also have intracellular calcium helps in the secretion of these hormones. Okay, and examples I have here are insulin, corticotropin, and growth hormone. A big word I want you to remember is signal transduction. I put this as a homework for you. And signal transduction is a process by which a hormone can bind to an external receptor like this and then stimulate a series of events within the cell, leading to greater events within the cell. So that is signal transduction. So hormones typically stimulate signal transduction. They send information from the outside of the cell to the inside of the cell through a series of effector proteins and second messengers. Okay, so that's how hormones typically work. So we're going to skip over this. It's so much more information than I want you to know. But here is a receptor kinase, tyrosine kinase receptor, which is an insulin receptor, leading to a whole lot of intracellular events, a whole bunch of intracellular events. So we can see insulin here stimulating mitogenesis, protein synthesis, glycogen synthesis, glucose transport, metabolism, and storage. See, this binding of a single hormone can stimulate many things in a cell. This makes the hormone pleiotropic. Pleiotropic. I want you to look up that word, pleiotropic. And the reason why a hormone can stimulate multiple effects within the cell is because of the presence of different intracellular proteins, effector proteins that can respond differentially to that same hormone. All right, let's stop there and we'll pick up in the next slide.